Hello and welcome to my presentation on the application and optimization of direct PCR in community metabarcoding. My name is Nina Roeder and I'm very happy to present at the first DNAQUA International Conference today. Traditional genetic barcoding approaches enable species identification through the application of DNA extraction, PCR and sequencing methods. However, direct PCR allows the amplification of DNA from tissue samples directly, without the need for DNA extraction or purification steps. The successful application of direct PCR largely depends on the absence of PCR inhibitors in the organism's tissue. With the emerging next-generation sequencing techniques, the simultaneous identification of many taxa became possible. Although direct PCR has been successfully applied in traditional barcoding approaches of invertebrates, it has not yet been attempted in community metabarcoding applications. In order to test if amplicons produced by direct PCR could be used for next-generation sequencing, we used the diverse community of non-binding midges, also called chironomids. Those had been sampled via emergence traps from an artificial pond mesocosm and stored in ethanol. The individuals were first dried, then homogenized, and finally dissolved in water. By pipetting the tissue-water mix, we combined 14 different chironomid species to a mock community of known composition. In order to compare direct PCR and traditional DNA isolation PCR, we used both approaches for amplification. After a second PCR for indexing and adapter ligation, samples were sequenced on an Illumina sequencer. We found only slightly lower recovery rates for mock communities with the direct PCR approach compared to traditional protocols. These recovery rates were further improved for both methods when chironomids were previously size sorted and an equal biomass of chironomid specimens was used. Generally, direct PCR did not have a large effect on sequence read abundance. However, read abundance varied strongly between species. Please mind the logarithmic scale on the y-axis. We are currently investigating whether this was caused by primer bias or an artifact of differently degraded tissue. In conclusion, we were able to prove that the amplicons produced by direct PCR can be used for next-generation sequencing, with possible applications for future biomonitoring projects and portable laboratory technologies. And with this, I would like to thank you for your attention.